I am Dr. Silvan, Assistant Professor, PG and Research Department of Biochemistry, St.Joseph College of Arts and Science, Autonomous. Now I am going to present the topic Post-Translational Modification. Post-translational modification is the chemical modification of a protein after its translation, or otherwise the chemical modification that takes place in certain amino acid residues after the protein is synthesized by translation are known as post-translational modification. These are essential for normal functioning of the protein. Post-transcriptional modification mostly occur in endoplasmic reticulum and Golgi apparatus. Why the post-translational modification is necessary? The post-translational modification is necessary for the stability of protein, biochemical activity, protein targeting, protein signaling. It means the protein to protein interaction and cascade amplification. The post-translational modification having a lot of steps are involved. The major steps are phosphorylation, lipidation, epicotination, disulfide bond, acetylation and glycosylation. The first one, N-terminal and C-terminal modification. All polypeptide begin with a residue of N-formyl methionine in bacteria or methionine in eukaryotes. However, the formyl group, the N-terminal methionine, must be removed enzymatically before they convert into functional protein. The process hydrolyzed by the enzyme deformylase. One or more N-terminal residue mainly removed by aminopeptidases. The amount of the eukaryotic process amino acid group N-terminal acetylated after translation. The next one, N-acetylation. N-acetylation means the addition of acetyl group to the nitrogen. Histones are acetylated on lysine residues in the N-terminal tail as a part of gene regulation. It's involved in the regulation of transcription factors, effector protein, molecular chaperones and cytoskeletal proteins. The major enzyme methionine aminopeptidase is an enzyme responsible for N-terminal acetylation. Next, the loss of signal sequences. Some 15 to 30 amino acid residues at the N-terminus, they a role in dissecting at the protein. This process to its ultimate destination in the cell, such sequences are ultimately removed by specific peptidases. Next, the covalent modification. The first one, phosphorylation. Phosphorylation means the addition of phosphate group to a protein, principally on serine, threonine or tyrosine residues or hydroxyl group, is also known as phosphoregulation. The critical role in cell cycle, cell growth, apoptosis and signal transduction pathways. The major reaction ATP plus protein, the action of the enzyme protein kinases to form phosphoprotein and ADP. The next one, hydroxylation. During hydroxylation, the collagen, the amino acid residues like proline and lysine, these are converted to hydroxylproline and hydroxylysine. lysine. The hydroxylation process majorly occur in endoplasmic reticulum in the require of vitamin C. The third, carboxylation. Carboxylation means the addition of carboxyl group, especially the aspartic acid and glutamic acid are carboxylated. The major examples for carboxylation enzyme is prothrombin. Methylation. Certain lysine residues are methylated enzymatically. Monomethyl and dimethyl lysine residue are present in muscle protein. In cytochrome C, calmodulin of most organisms contain one trimethyl lysine residue at specific position. The another protein glucose residues undergo methylation. Disulfide bonding. Disulfide bonds are covalent bonds formed between two cysteine residues. These bonds contribute to the correct folding of proteins as other elements of secondary structure. Glycosylation. The glycosylation, the covalent attachment of oligosaccharides. The addition of glycosyl group or carbohydrate group to a protein, principally asparagine, hydroxylysine, serine or threonine. The significant effect of protein folding, conformation, distribution, stability and activity. Addition of prosthetic group. Many prokaryotic and eukaryotic proteins are actively covalently bound to prosthetic group. These group become attached to the polypeptide chain after leave the ribosomes. Examples acetyl-CoA carboxylase and heme group like cytochrome C. Addition of isoprenyl group. The addition of isoprenyl group to the cysteine residues of a protein. The isoprenyl group are derived from pyrophosphate intermediate of the cholesterol biosynthetic pathway such as farnesyl pyrophosphate. The last one, proteolytic trimming. Many proteins like insulin, glucagon and the proteases like trypsin and chymotrypsin are initially synthesized as larger inactive precursor. These are proteolytically trimmed to produce a active form.